researchers in PKU. I have people who've been working on PKU for more than a decade longer than I've been alive. And so it's a really interesting group. I joined Synlogic a year ago because I was interested in working more actively on developing therapies. And what I'm really here today to talk about is the PKU therapy, but also a little bit about our HCU program. And I did it because I was interested in getting to work on introducing therapy faster. The company I joined, Synlogic, I really joined because their technology is kind of cool and different. We are trying to do something that hasn't really been done before. Our medicines are actually living organisms. They're engineered bacteria that are based on a probiotic that people have been taking for many, many years. That probiotic is widely available in Europe, and people take it for digestive health. It has limited availability in Canada, and it's of course not available in the United States at all. It's impossible today to improve. We have taken that organism and re-engineered it to do a couple of tricks for us. It actually imports phenylalanine from the food that you eat, and it degrades that phenylalanine using enzymatic systems. The product is in progress. Your stomach and the upper part of the small intestine to create phenylalanine, and then goes away harmless. Some of the questions that we often get are, why can't you just give this organism and let it survive in patients to continue working long term? Why can't we do one-time therapy? The problem there is that your microbiome lives in the lower part of your intestine, and that's not where phenylalanine. So it actually wouldn't work as a strategy. The other reason has to do with moving Jurassic Park, if you remember that, which is you can come up with all sorts of strategies that you think will prevent the living organisms from dividing, and you're not entirely sure that this will work. But in our early phase studies, we showed that the strategy is actually quite effective. So I think I'm up on screen, so I'll start making some of these slides. I'm going to skip through a bunch of them, and feel free to stop me and ask questions from the time. I want to start with the disclosure that of course I'm an employee at Synlogic. And I basically talked about most of this. I'm going to skip through it. This is actually the product that's sold in Europe and to a limited extent in Canada. Mutafor, which is the underlying organism that we use and have re-engineered to become our medicine. And it was actually isolated from soldiers in World War I. So the medicine is actually basically ancient in origin, and was used because soldiers had trouble with gastrointestinal health during the war, and it was thought that the use of this product could prevent them from having cholera. The understanding of cholera was somewhat imperfect during World War I. It's all right.
While he's doing that, I have a question.
Yeah.
If, sorry, I don't What was that called? Beautiful?
Beautiful is the.
You said it's really prevalent in Europe, but
Yeah.
not true here.
It actually is approved in Canada.
Oh, okay.
You can get it. I found it very difficult to find. It is not approved in the United States.
Oh,
Yeah. I've gone into health food stores in Toronto. Sometimes I found it, sometimes I haven't.
how does it differ from just a probiotic that you buy at Costco?
It's a single organism probiotic.
Oh,
So probiotics come in many different flavors. And you know, yogurt itself is basically probiotic.
yeah.
It's actually an organism. Beautiful is just a single type of bacteria. And these are all helpful bacteria. These are the type of bacteria.
I just, like, I see the word E. coli and I'm like,
So we have at any given time more than 1 trillion E. coli cells living in us all. So it's not each and every one of us. So we're all colonized by bacteria all the time. People don't like to think about it sometimes, but it's true that we live, covered with bacteria on our skin. But here I'm talking about the bacteria that live in your intestine. And you actually need the microbiome. It's quite unhealthy not to have a microbiome. They aid in your digestion. And so if you actually injure your microbiome by taking antibiotics, you can actually have problems like diarrhea and constipation as a result of it. So our medication requires people not to be terribly squeamish about the prospect of taking E. coli, but we have E. coli in us all the time.
I think there are bad forms of E. coli and good forms of E. coli.
There are bad.
Is that an oversimplification?
That is not an oversimplification. That is actually true. There are forms of E. coli that can cause illness and actually cause food poisoning. But there are many forms of E. coli that are harmless. This is just an example of one of the enzymatic systems, and the reason that I point this out is that one of the enzyme systems that we use, that's probably the workhorse of our medicine, is actually the same that's used in Palynziq, or pegvaliase. It's called phenylalanine ammonulase. That's a medication that was recently approved in Canada, although it isn't widely available. And the difference between our approach to using phenylalanine ammonulase and the approach used by Palynziq is simply where we're giving it. So in our case, we're giving it enterally, so it doesn't actually enter your system. And this can avoid some of the potential side effects. Palynziq is given by injection and works basically in the bloodstream. And so there are relative advantages of disadvantages to those two approaches. The advantage to giving it in the gut is that you're less likely to develop the immune symptoms that have been a problem for people who use Palynziq. And the challenge for us is we have to give it orally and fairly continuously for each month. So the product is meant to be given.
you said, like, so how long does this bacteria actually survive in so many decades?
So it's actually an interesting question about how bacteria are alive. You could think of the bacteria that we are using as our product as being dead at the time we give it to you, because actually the definition of being alive for a bacteria is whether it can divide or not. And since our bacteria is non-dividing, you could think of it as being dead at the time.
Actually, how long does it stay inside?
So less than 2 weeks is sort of all that we know. So in our early phase studies, we actually had to take stool samples from people and do polymerase chain reaction studies to show that the organism is gone. And it's gone after 2 weeks. And that was the earliest time point that we looked at. So we think it basically goes through within days, but it doesn't divide, so it's difficult to say whether it's sort of alive or not. But it's sort of an interesting philosophical question of bacteria and philosophy. Again, I'm gonna skip a bunch of these slides, sort of get on to a little bit about talking about the clinical trials. So this is just a list of the studies that we've done to date. We have to do many types of studies. In the beginning, we give these medicines to healthy volunteers, and then in phase 2 studies, we give it to patients. I'm sorry, I'm not being loud enough, apparently. Not usually a complaint that I'm given, but in phase 2 studies, we give the product to patients. I've had a surprisingly recent opportunity to discuss phase 2 studies today. Our phase 2 study has been completed and is about to be published. Phase 2 studies are fairly small studies in terms of the number of patients and look to see if the medication can safely lower phenylalanine. I'm actually going to skip a few of these as well. This was just the design of our phase 2 study. And I want to actually focus on this, although it might be a little bit hard to focus. One of the challenges that we have actually has been gastrointestinal adverse events. Feeling sick to your stomach when you take the product. And a lot of what we've done in between phase 2 and phase 3 is work to make that better. In my very recent experience discussing the phase 2 with someone who did it was pointed out that one of the problems we actually had, and we were told this, is that the flavoring agent that we used was too strong, it bothered people, and we've actually changed that. We've also done other things to improve the palatability of the medication. Probiotics can be easy for some people to take and can be difficult for others to take. The product is actually a powder, which is mixed in 100 cc's of liquid and taken right after meals. And it can be taken with either water or apple juice. And we've done what we can to improve the flavor of it to make it more tolerable. PKU patients, unfortunately, are used to taking things that don't always taste the best. So in a sense, they're sort of an easy audience to do these kinds of things in. But we want to make it so that there's a great advantage to taking this product compared to taking medical formulas. So discontinuations from the study were one of our biggest challenges in the phase 2. The medication works, and this is sort of the data we use to show how the medication works. This is when we give phenylalanine to patients in the study, and we look to see how quickly it clears. It shows the amount of lowering of phenylalanine uptake from the gut. And we see with the product that we're bringing into phase 3 that there's about a 40% reduction in phenylalanine transport when we give the medication. And that correlates to the level of phenylalanine reduction, the plasma reduction that we see in patients in the phase 2 study. What I really want to talk about today, because we're a little short on time, is actually the phase 3 study. The phase 3 study has started. It actually is starting up in Canada somewhat more quickly than it's starting up in the United States, maybe because I'm Canadian and I run clinical development, maybe because I love to argue about Canadian regulations, but actually Health Canada is great to work with. From a pharma perspective, they're lovely. They work very hard and they work very efficiently, so it's easy to set up trials in Canada. We have 2 active sites currently. One's in Calgary, one's in Hamilton. And we will eventually, we think, have 6 sites in Canada to enroll this study. We're currently enrolling only adults because the regulatory agencies wanted us to work with adults first. We hope to work with adolescents sometime during this phase 3 study. We will have to work with younger children, so children under 12, after this study is completed. And that's typical for drug development. You're not allowed to work with kids first unless the disease kind of only affects kids. In the study, one of the things we're doing to make the medication more tolerable is we're saying, okay, not all patients are going to take the same dose of this medication. And we're asking people to step up in taking the medication to a level that they're comfortable with. Once we find that dose in what's called the dose escalation period, we do what's called a randomized withdrawal, where we take half of the people off the medication and give them a substitute placebo that looks exactly the same. That's the phase of the study where we actually collect our data. Yeah.
What's the criteria to be comfortable? You said we
That
take them
is-
up to be comfortable.
Yeah, that's something that's going to be determined by the individual investigators and patients. So they'll be told they have the option to increase the dose that they're taking. Do they intend to increase the dose that they're taking or not?
So it could be based on the side effects the individual's having.
That's right. That's right. We want to be sure that people are taking amounts that are right for them. And part of the reason is that's how probiotics work. Probiotics are not taken the same way by every person. They're taken at different dose levels by different people based on their tolerance. We're interested in treating people regardless of how much of the product they use. We do think that the more of it you take, the stronger the effect will be on phenylalanine level, but we think there's a value to it at multiple different levels. Yeah.
Just curious about the increase in dosage. So would you be increasing the volume?
No, the volume stays the
Okay.
same. In fact, it won't look any different. The packets they're different colors to keep people from confusing them. But the amount of powder that you're dissolving looks exactly the same. Regardless. Yeah.
You may have mentioned this, but do you expand this to other conditions as well? Like for
We do. So, and actually, I'll skip to this slide. So the other program that we're working in is on homocystinuria. So we've completed a phase 1 study of homocystinuria. In homocystinuria, obviously getting rid of phenylalanine doesn't have any value. There you're targeting methionine, which is what's converted into homocysteine. And it's interesting here to be talking to CanPKU plus because I know that that's actually a focus, and we're going to be hearing about homocystinuria as well. But homocystinuria is a disorder that has a very different set of complications, but has similar dietary principles. Again, one of the things that attracted me to working at this company is this is actually a modular system. There are many metabolic disorders that I take care of where there is a diet where something is dropped out. And you can actually design enzymatic systems to get rid of other amino acids or other offending substances, which are the basis of care for a disorder like maple syrup urine disease, which is a disorder where leucine amino acid is a problem. In homocystinuria, we've only done phase 1 studies, so we haven't treated patients yet. Homocystinuria is a somewhat more difficult disorder to tackle in some ways because there are many fewer patients with HCU. So at Sick Kids, I take care of all the HCU patients and all of the PKU patients, and I have about 90 PKU patients and 2 HCU.
What about ammonia?
What's that?
Ammonia, is that something you're looking at?
So we actually did early on look at treatment of urea cycle disorders, but we were unable to design an enzymatic system so far that we wanted to take forward to clinical development. So we would need to have a way of fixing nitrogen to be effective to treat urea cycle disorders, and we're going to have a talk about UCDs. Okay, that's what I recognize. You know, they're taking care of the child. Yes. Yeah.
Yes, I have a baby boy.
Yes, and I take care of it.
You taking care of it?
Yes, I'm one of the staff physicians at Sick.
Okay.
So again, it's a modular system, and we can take care of other disorders. And sorry, this has been a somewhat disorganized talk, but I'm happy to take any questions about the program. We are enrolling. If you know of people or have interest in yourself in participating, over at the booth, I have a little flyer that can give you information to get connected to sites to participate in the trial. If you're interested, I'll open one of those up right now. And I'm happy to answer any questions people have. And I really appreciate it.
How many adult patients do you need for phase 3?
We need 150 patients for phase 3. We don't have all 150 since we just started. So we are certainly looking for more patients. And again, the trial lasts 3 years in total, including the open label extension. The open label extension is actually quite an easy phase of the trial where people just continue to take the medication. The key early phases take anywhere between 2 and 4 months, depending on the dose that you go to.
Will this be, so for any folks that are enrolled in the study and have success, because it's a study, the drug's not FDA approved or, so.
Well, Health Canada approved is the important list, correct?
Yeah, exactly. So would the individuals be eligible to have the drug, like continue with it as a compassionate benefit?
So we use the open label extension as a start to that process. We can never promise the availability of it because we have to show that it's helpful.
Okay.
So we have to do the study first to show that it's helpful to allow compassionate use.
Okay.
Drug companies have traditionally used compassionate use. Canada is a lovely place, and John can tell you a lot about this, to do research, kind of a tough place to get paid back for your drug sometimes. So many companies have used the compassionate use program to get around that and allow patients who participated to continue to use the product while obtained in a rate that is coming out. But to do that, we have to be sure that the drug is working.
Okay.
And that's what a phase 3 study does, is it proves that the drug is effective in actual patients.
Are they looking for like a majority of folks that respond and successful, or?
So we have very complicated statistical tools that I can bore you with that are what we use to prove to governments that the drugs work. But basically, we use an analysis of phenylalanine lowering in patients to show that the drug is effective. No drug is effective for every single person. That's not how drugs work. We have to show that it's effective for the population of patients that we're studying.
Do genetics play a portion in the success of?
No.
No.
So unlike Karen medications like sapropterin, CBFterin, Humana, this medication can work for any genotype because it acts on phenylalanine before it gets to your endogenous system for clearing. So some of you in this audience have PKU that allows some pH activity, and that responds to medications like Humana. And others have the type that really you don't have any pH activity at all. And terrans can't help people like that because that's not what terrans do. This medication would work for anybody, and that's true also for palatin, to be clear, because we're not relying on the endogenous system.
So in the past phase studies, do, for the medication, so physiologically it's effective in the gut, so do most people that don't continue to use it, or is it more from the side effects experience?
That's what happened so a phase 2 study is not supposed to be a continuous study, it never is. So the study ended for all the patients, even those who did well on it.
Oh.
We aren't allowed to continue to give medication to people in phase 2. But the patients who discontinued generally had sinus problems. One person had what appeared to be a food allergy.
Oh.
And we don't know what they were reacting to. It could have been the flavoring agent, it could have been our product, it would only happen once, so we're not sure.
Or did they expand to something their diet that they?
We asked them not to.
I'm conscious of time.
Oh,
And just quick, 2 points very briefly.
Yes.
How long are you with us today?
I am with you until my flight leaves in the afternoon.
Okay, so you're going to be around at the lunch break, whatever, to do follow-up. And you're way too shy. Your most important qualification you failed to mention, that you and your family moved from the United States to Canada, and this year you and your entire family became Canadian citizens.
We are.
All right, yeah.
And then I guess I do have, will you share your PowerPoint? Because we flipped through it so quickly, there's lots of it that's.
I've attempted to, I hope I can get it to CanPKU somehow.
Okay, that'd be great, thank you.
We'll do our best, we'll get it, yeah. And we'll post it on our website. Okay, so,
That's great.
Yes, sir.
okay, thank you so much. Some of you may know my son went through, in 2007, joined the phase 3 clinical trial for the drug that became Kuvan. So I've kind of lived
The next
learning
Zoom.
experience of participating in the phase 3.
So
Actually,
you hit the next Zoom, which will be the,
but I'm happy to have a conversation.
this one, yeah.
Talk about the ins and outs of that.
All right, while she's getting set up, there are 2 things I'm not going to attempt to pronounce in this introduction. And number 1 is Kate's last name, and the other one is the actual name of this disorder.
It's in the agenda.
Kate here is going to do a presentation now on what's called OPC, and no, I'm not going to try and say it.